guys, my name is Samantha. I'm a fourth year biomedical science student at Ryerson University, and I'm currently doing my senior thesis in the lab, as well as volunteering in a bunch of different student groups. So I'm excited to show you what a day in my life looks like. Hey everyone, so as mentioned, my name is Samantha, and I am currently doing my senior thesis at Ryerson University. And right now I am in my morning lecture. It's 8 a.m. in the morning. I have a 2-hour cancer bio lecture, and this is sort of my setup for online learning, and I find it works really well. I have the double monitors because it allows me to put the lecture and the Zoom call on one screen while having my notes on another, and this sort of emulates how it would be in person as well. And so what I do throughout the lecture is I type notes that correspond with the slides that they provide us, and then later on, this is the same as in person, is I'll handwrite those notes out as well. This really helps with memorizations and really putting the information and absorbing it for exams and assignments. And so basically I'm just following along with the videos and lectures, and here I'm writing in my agenda, and that just helps me keep track of all the stuff I have to do that week. I also have Google Calendar, and I use that to sort of schedule my weeks. And after this class, I actually had a meeting with the course union, and so as mentioned, I'm involved in a bunch of different student groups, and the course union being one of those, and I really recommend getting involved in student life when you do make that journey to post-secondary education. It's very fulfilling. But yeah, this is just my setup. I do really like it, and I'll hopefully transition. And so part of my day involves commuting to school. So Ryerson is sort of known as the commuter school. I take the GO train in every day to go to the lab. And then when I get to the lab, I start off with just writing in my notebook what I need to get done today, sort of writing down any notes I need to do, any calculations, and setting up from there. And I sort of try to plan out ahead of time what I need to get done. So today, I'm going to be doing a bunch of different stuff, as you'll see. But first off, I just want to talk a bit about my project. And so my project actually looks at histone chaperones, which are essentially guide proteins that help chromatin, which is how your DNA fits in your cell, basically form. So it helps like DNA come super condensed. Maybe you're familiar with the cells, and there's X chromosomes that you could see during mitosis and stuff like that. So that's essentially how histone chaperones help those structures get formed. And so we're looking to see if when they're not available, or when they're deleted or mutated, if the cells are still able to grow and proliferate. And the reason is because this could be a cancer-causing mechanism, and we're especially looking to see if it's able to continue on through the cell cycle when we sort of introduce DNA damage. And DNA damage is how mutations evolve. And this is sort of coming back to that idea of cancer research, and understanding these mechanisms can actually help us understand how cancer, through epigenetics or DNA mutations, arise. And so here I am, I was labeling my tubes before, and now I am filling them with water, and right now I'm going to be staining samples from a previous experiment. That way I can visualize it on a microscope on a later date. And so part of that is just filling and vortexing. Vortexing is just a way of mixing samples and any other liquids into a homogeneous solution. And so that's just what I'm doing here. And it's a long and tedious task, and lots of pipetting. I think there are about 32 samples I had to do today. So now that I'm done doing the samples for staining, I'm putting them in the centrifuge so we end up with a pellet at the bottom. And that's just the concentrated form of the strains we're using and their cells. And then I'm pouring out the excess liquid. That way we only have that. There's no extra water or ethanol in that tube that I need for staining. And so now I am adding more water to the cells, and I will go and get the staining. And so we use aniline blue and DAPI to stain our cells. So the aniline blue, it just stains the cells blue and allows for us to actually just visualize the cell altogether. Now the DAPI in specific allows us to visualize the nucleus and the septum. And now the septum is only there after mitosis. And so mitosis is when the cell is getting ready to split off into two. And so when the septum forms, essentially it's where the cell will break apart into two daughter cells. And so I just finished doing the DAPI staining for those ones, and so I went to go put the tubes into the fridge. And now moving on to the next experiment. So this is actually something I've been doing a lot of, and it's testing the sensitivity of our different samples and strains. So the ones that have like maybe histone deletions, and sometimes we did cell cycle deletions. But we're just testing its sensitivity to DNA damaging drugs to see if when there's certain genes unavailable to the cells, can they still go through the cell cycle in the presence of something that causes stress. And so right now I'm making my overnight cultures, and these will just sit on my bench top and be ready for the next day. And so what I'm taking them from is stuff called wake ups, and they're just fresh grown strains on a agar plate, which is just a medium that helps cells grow and gives them nutrients. And so now I'm actually doing a cross, and a cross is when you take two different strains. So maybe you have a wild type and you have a mutant, and you want to cross them to see if there's an additive effect or a negative effect or something else going on, if they're viable. And so I just did that. So now it's actually the next day, because I did want to show you part 2. And so especially for the drug sensitivity. And here I am, I'm getting my cultures. I had left them to incubate for an hour and a half earlier in our air shaker. And I have my plate, and I'm getting my pipette set up. And in that pink little rack, there's actually concentrated drugs. And so in this case, I'm using hydroxyurea, which is a DNA damaging drug, or replication stress drug, sorry. And that'll be added first to our assay. And so the assay is actually contained in a 96 well plate. And as you can see here, I'm just pipetting into each well. And yes, I did pipette into all 96 wells twice, because I did 10 microliters of the drugs, and we did different concentrations so we could see if there was increased effects or if there were decreased effects, depending on how much drug was available. And then here I am, and I'm starting to pipette in the samples, and each sample is contained in a certain roll. So we had about 8 samples, and we did 4 different concentrations, and each one was in triplicates. And so triplicates just means we replicated it 3 times. So there's 3 wells for each concentration and each strain. And so it doesn't take too long to do this. You know, pipetting is something that you gain as a skill in your undergraduate labs, and even more so in the thesis lab. And then when I'm done, all I did was put it into the machine, and then it reads for 24 hours. So now I'm on my way home, and when I get home, I work out right away, as soon as I get home. It's a nice way to de-stress for me. And, you know, I have a home gym in the basement, as you can see. Yeah, and I just really enjoy working out, and I really promote students having some sort of hobby on the side, because school, especially in the STEM fields, can be a lot and stressful. But yeah, that's pretty much my day as a student and in the lab as a researcher through my undergraduate thesis. And it was fun to take you along on that, and I hope it provided some insight. And yeah, it's quite a fun journey. It can be stressful at times, I'm not gonna lie. But I think research is a huge component of an undergraduate within the STEM field. And I think everyone should have the opportunity to get into the lab when necessary. Obviously, not research is not for everybody, but if you're considering it, it's definitely fulfilling and it's very interesting. And get into an area you like, because you could have a project and you have no interest in the project, and then it just feels like so much more work. Where the project I'm doing right now, I'm very invested in it and I really enjoy it, and so it really feels like a hobby more than anything. But yeah, and that's my day as a student researcher. Hope you enjoyed it.